Assalamu alaikum, everyone. My name is Immad Ashraf. I'm a second year medical student, and today we'll be going over large intestinal motility, which is a lecture in GIT physiology. It's one of the easier lectures, so it shouldn't take too long, and it's a lot of common sense. And if you've done the previous lectures, then a lot of the concepts are already in your brain, so it shouldn't be too hard, inshallah. If you do not feel like asking any questions during the lecture, you can ask them after. You can send me a message or email me. So let's get started. Bismillah. So we're going to start with this diagram that shows us the colonic segments and their contractions. So a segment of the colon is called a haustra. So all of these segments that you see that are separated, for example, wait, let me just change the color real quick. For example, this, or this, or this, these are all haustras, and that's spelled like this, haustra. So haustra is a segment of the colon, and these aren't permanent, so they're dynamic. They come and go, and this is due to the fact that the haustras are formed by segmental contractions of smooth muscle. So the smooth muscle present in the intestine, once it contracts, it forms a haustra. And when it relaxes, this haustra is no longer there. That's why we call the haustras dynamic, because they're not present permanently. So what forms the haustra really is that, just imagine with me that this is a part of the large intestine. Now, when the smooth muscle present in the large intestine contracts, it contracts something like this. Okay, so here's one contraction, and then, so it's contracted now. And this is one haustra, this is another, and this is a third. So since it's contracted on either side, it leaves this small opening in the middle, and this is called a haustra. So the contractions now, those were the segments, and now the contractions are non-propagating. Most of them in the colon are non-propagating. And what does that mean? So there's two types of contractions in the GIT in general, propagating and non-propagating. So non-propagating are segmental contractions, which means it doesn't move the feces, which is present in the large intestine. It doesn't move. So once again, this is the large intestine, and let's imagine this is the direction towards the anus eventually. So if it's moving, then it's propagating. The contraction is propagating, moving. But if it's non-propagating, such as in most cases, that means it isn't moving. It's just present in one haustra, and it's moving within that haustra and getting mixed. So I hope you understand that, but I'll repeat it again. So non-propagating means it isn't moving towards the anus. It isn't moving. It's just stationary in a particular haustra, and it's getting mixed so that the nutrients can be absorbed. Not the nutrients, but mostly the water and the sodium, but also some nutrients. Whereas in propagating contractions, there is movement of the feces towards the anus. So most colonic contractions are non-propagating, which is similar to the segmentation contractions in the small bowel, in the small intestine. So why do we have contractions? As I said, it's to mix the contents of the large bowel and enhance exposure of fecal material to the mucosal surface of the large intestine so that there is maximum absorptions of fluid and solute. So the non-propagating contractions obviously mix the contents because they're not going anywhere, they're just getting mixed. And it also enhances the exposure to the mucosal surface for absorption. So these contractions, what controls their frequency and timing? So basically, what controls these contractions? And the answer is similar to all the other contractions that are present in the GIT. It's via the intrinsic slow wave activity or the basal electrical rhythm. So we have the basal electrical rhythm, which is controlled by a pacemaker, in the GIT, and this pacemaker has these electrical signals that are converted into these intrinsic slow waves, and then these slow waves are what cause the non-propagating contractions in the colon. These are also present in the small intestine and in the stomach, which you must have studied in the previous lectures. Okay, so we're done with that slide. Once again, I'm going to keep repeating this, but Hofstra are dynamic, so they come and go. They're not permanent. They only come when the large intestine smooth muscle contracts. So and then again, I made sure that you guys know the difference between propagating and non-propagating, because it is a bit confusing. So non-propagating. So the complete definition, as per Google, is non-propagating colonic contractions refer to contractions of the colon that do not propagate or move along the length of the colon. So there's no movement along the length. These contractions are known as segmental contractions or haustral contractions, okay? Because they're within one haustra. Remember, a haustra is this little pocket that's formed when the smooth muscles contract on either side. And why do they not propagate? So in non-propagating colonic contractions, why is there no propagation? Because the lumen is blocked on either side. So there's contraction over here, which means it can't move backwards. And there's contraction over here, which means it can move forward. So all it can do is be mixed up in this haustrum. Haustrum is the singular form of haustra. So all it can do is get mixed up in this haustrum. And this causes obviously the mixing and eventually the absorption. Okay, so now we have propulsion and retropulsion. So in general, propulsion means moving forward and retropulsion means moving backwards, right? So we have haustral propulsion, which is when, okay, so once again, let's imagine that this is the large intestine without any contractions. And then let's imagine that the smooth muscle contracts and forms these haustras, right? I'll just draw them like this, okay? So now we have three haustras over here. So in propulsion, adjacent haustra contract in sequence. In sequence means one after the other in an ordered manner. So contents of a single haustrum are displaced into adjacent haustrum and continue downstream. So let's imagine that this is the direction towards the anus. And then these are three haustra. Obviously, in the intestine, we have a large number of haustrum, but just for explaining, let's imagine there are three, right? So adjacent haustra, which means let's label this 1 and 2, contract in sequence. In sequence means in an order. So let's say, in this case, 1 is going to contract, okay, this is going to contract, and push its feces, or whatever is present in the lumen, into 2, okay? And 2 is going to relax, to allow 1 to push the feces into it. So now that the feces are present in 2, the same thing is going to happen with haustrum number 3. 2 will contract and 3 will relax, and the feces will move into 3. And this continues until we reach the anus. And that's called haustral propulsion. Propulsion because it's moving forward to exit the body. Haustral retropulsion is the same thing, but in the opposite direction. So in this case, we're going from 1 to 2 to 3, eventually to reach the anus. But in this case, we're going from 3, so in retropulsion, we're going from 3 to 2 to 1. So we're going in the other direction. It's still sequential, but the sequence is in the opposite order than in haustral propulsion. So this moves the fecal material orally. So it doesn't move it towards the anus, such as like propulsion did. It moves it towards the oral cavity. Not obviously all the way to the oral cavity, but that just means in general in this direction. Okay? Retropulsion means moving it backwards. So in this case, 3 would contract and 2 would relax, and the food would move backwards. Not the food, the feces. The feces would move backwards, and then 2 would contract and 1 would relax, and then it would move from 3 to 2 to 1. And then we also have something called multi-haustral propulsion. So in multi-haustral propulsion, we have simultaneous contraction of several haustra. So instead of a single haustra contracting and then another one relaxing so that it can move, many haustra that are adjacent to each other will contract and then empty their content into the adjacent haustra downstream. So let's say we have 1, 2, 3, So this time, let's say we have 1, 2, 3, 4, 5, 6 Haustra. Okay. I'm sorry, the diagram isn't really good, but just let's imagine there's 1, 2, 3, 4, 5, 6. So if we have multi-Haustra propulsion, what happens is that 1, 2, and 3 will contract, and 4, 5, and 6 will relax. And all of the feces that is present in 1, 2, and 3 will empty into 4, 5, and 6. So it's just a faster and more efficient method of Haustra propulsion. And that's why it's called multi-Haustra propulsion, because many Haustrum are involved. So Haustra contractions occur with increasing frequency after a meal. So once you eat, the Haustra contractions are going to increase in frequency. That's pretty self-explanatory. That's it for this slide. And then this is just further explanation. So the first thing is, why are Haustra contractions slow? Because I mentioned that the Haustra contractions must be slow so that we can have the mixing in a single Haustrum, and we can have the absorption of electrolytes, water, and nutrients, right? So the answer to this is due to the lack of the basal electrical rhythm gradient. So we have a lack of the gradient. So the slow wave frequency, which is obviously due to the basal electrical rhythm, is variable in the colon and not characterized by proximal to distal frequency gradient. So usually what happens in other parts of the gastrointestinal system is that the basal electrical rhythm is highest proximal and then least distal. This allows for a flow. So let's say this is proximal and this is distal. The basal electrical rhythm would cause the contractions in such a manner that that whatever the content was in the lumen would move distally. But this is not the case in the colon. They're not proximal to it doesn't decrease from proximal to distal. In this case, it's erratic, so it's different. This is distal, this is the anus, and this is proximal. Okay, so you can see proximal, it's first of all, it's 7 to 8 in the ascending colon. It's less than 11 at this flexure over here, and then in the transverse colon, it's 11, and then it continues 11 in the descending colon, and then in the sigmoid colon, it's again less than 11. But then in the rectum, it's 17. You don't need to remember any of these numbers. These are just to show you that it's erratic. It is not a single number, and it doesn't increase or decrease. It's literally random numbers. And the highest BER is in the rectum. It's 17 per minute. So the erratic BER is the reason for the erratic movement of the chyme or the feces through the colon. So it doesn't follow the decrease from proximal to distal like all the other parts of the GIT. Instead, it's random, it's erratic, and obviously this helps in the mixing. But this is the reason why. This is also the reason why the contractions are slow, because there's no real gradient. As you can see, it increases and then it decreases and then it increases again. So that's it for this slide. So now we have the different contractile activities, but before that, if anyone has any doubts, you can ask me right now. You can unmute, or you can put it in the chat. I can't see the chat, so actually it would be better if you unmute. So if anyone has anything to ask, or you want me to repeat something, you can unmute and ask me now. Okay, I guess no one has any doubts, so we'll continue. So the contractile activity in different segments of the colon is actually different. So as I said, in most of the colon, it's non-propagating, right? But there's also some parts of the colon that have propagating movements. So let's talk about the cecum and the ascending colon. By the way, I'm assuming that all of you know the basic anatomy of the large intestine. We have the cecum, which is like the first part. Then we have the ascending colon. Then we have the flexure. Then we have transverse colon. Then we have a flexure, and then we have the descending colon, then the sigmoid colon, then the rectum and the anus. So the cecum and ascending colon are the major sites of fluid and electrolyte absorption. So these are the major points at which the fluid and electrolytes are absorbed in the large intestine. The movements are non-propagating, which is similar to what we learned before. That means they're segmental. So which means that there is contraction within the haustrum. So it's also known as haustral contraction, and there's mixing within the haustrum. And as you can see, it says the main motor activity is haustral shuttling, which is just the contraction of the haustrum, so that there's mixing and so that there's absorption. This happens in the cecum and ascending colon. In the transverse colon, we have the propagating contractions. What are propagating contractions? I told you there's two types, non-propagating and propagating. Non-propagating means there's no movement along the length of the colon, but propagating contractions means there is movement. And why are these propagating? It's because of the BER. The BER is higher than either proximal or distal segments, which means the fecal material can move along the length, either forward or backward. That doesn't matter. Propagating means either direction. So fecal material moves orally, which basically in the GIT means backwards because the oral cavity is the first point of the GIT, and aborally. Aborally basically means opposite to oral, also known as towards the anus, right? So as I said here, 11 per minute. You don't need to remember the number, just so that I can explain further. You see here, it's 7 to 8 per minute, approximately. And then over here, it's less than 11 per minute. So comparatively in the transverse colon, it's higher. And this is one reason why the movement here is propagating and not non-propagating. And obviously, it can move in both directions, orally and aborally. Luminal material that is propelled backwards from the transverse colon to the cecum mixes with fresh material that passes into the cecum via the gastroileal reflex, which basically means that when the food, or not the food, the fecal material moves orally, which means backwards in this case, it'll be mixed with fresh material that passes into the cecum via the gastroileal reflex. So the cecum is the first part of the large intestine. And we obviously have a chyme moving from the small intestine into the cecum. So when this moves backwards from the transverse colon, it's mixed with fresh material that has just come from the small intestine via the gastroileal reflex. I think you might have studied the gastroileal reflex in earlier lectures, but I still mentioned it here. And I have a full definition of it over here. So before we get to the gastroileal reflex, we must ask ourselves, okay, so the fecal material can go from the transverse colon backwards. Okay, it can go backwards to the cecum, but can it move backwards from the cecum to the ileum, which is the last part of the small intestine? And the answer is no. And why? It's because of the ileocecal sphincter, which allows movement only in one direction, right? It's closed otherwise. Now, the gastroileal reflex facilitates the movement of chyme from the stomach to the small intestine by relaxing the ileocecal valve. Now, you may ask, okay, so the movement of chyme from the stomach to the small intestine, what does the ileocecal valve have to do with this? The ileocecal valve is the valve between the ileum, which is in the small intestine, okay, small intestine, for SI, for small intestine, and the cecum in the large intestine. So what does the stomach have to do with this? And the answer to this is that it's an indirect effect. So when the ileocecal valve relaxes, it allows for the movement of whatever chyme is present in the small intestine into the large intestine. And this frees up the small intestine. So now, since the chyme has moved from the small intestine is empty. And because of this, we can add, the stomach can send further stuff into the small intestine because it's empty now. And the relaxation of the ileocecal valve is actually done via a hormone called gastrin, which is released in the stomach, which I'm sure you guys must have studied in the lectures about gastric secretion. So the gastrin causes relaxation of the ileocecal valve, which causes the movement of chyme from the small intestine into the large intestine. This causes the small intestine to be emptied, and now further chyme from the stomach can move into the small intestine. So I hope you understand now how the gastroileal reflex plays into this whole thing. So the gastroileal reflex causes the fresh material to move into the cecum because gastrin relaxes the ileocecal valve. Okay, so the net effect that happens postprandially which means after food. Prandial means eating or food. In the proximal colon. So what happens after you eat in the proximal colon? So you retain and mix. Sorry, this isn't supposed to say MIC, it's MIX. Retain and mix luminal contents and prolong the time available for absorption. Because in the proximal colon, you have which type of contraction? You have segmental contraction, also known as haustral contraction, or also known as non-propagating contraction. This contraction obviously causes mixing and absorption of food. So in the proximal colon, you have segmental contraction, which prolongs the time available for absorption of food. I hope that's clear. Now we're done with the proximal portion. Let's come to the distal portion of the colon. So the distal portion of the transverse colon, this is still the transverse colon. Previously, we were talking about the proximal portion of the transverse colon. This over here is proximal. Although there isn't much of a difference, but still, just to make sure you guys understand, this is proximal. And over here we have distal, right? So the propulsive activity, so it's propulsive or propagating, same thing, which results in the movement of colonic content towards the rectum. So it's propagating, which means food, or in this case, feces, moves along the length of the colon because it's propagating, not non-propagating, right? It's propagating. It moves towards the rectum to eventually be excreted. Usually, the movement of fecal material towards the rectum is slow. However, in cases of mass movement, it is rapid. So let's just keep the sentence over here because we're going to have a few slides about mass movement in a bit, and I'll explain this over there. So when the fecal material reaches the rectum, so now it's passed through the cecum, the ascending colon, the flexures, the transverse colon, another flexure, the descending colon, the sigmoid colon, and it reached the rectum, okay? It distends the rectum, basically it expands the rectum, and this causes the defecation reflex to be activated, okay? However, this doesn't mean we defecate, because if that was the case, then whenever our rectum is distended, no matter where we are, we would defecate, right? And that's not ideal. So defecation can be voluntarily suppressed. And if it is suppressed, then it is stored or moved back towards the sigmoid colon. Okay, so if it's stored in the rectum, we call this accommodation. So basically, the rectum accommodates the feces until we are in a position to defecate, because defecation is voluntary at the end of the day, to a certain point, anyways. Or it can be moved back towards the sigmoid colon and then stored there. And this is called retroperistalsis. So obviously, all of you know what peristalsis is. And if you don't, I'll have the definition of peristalsis in the next slide, I think, so we'll go over it over there. But peristalsis is usually in one direction, right? Like towards the anus. But if we voluntarily suppress the defecation, then the chyme, or the feces in this case, is going to move in the opposite direction, and that's called retroperistalsis. Retro for backwards, right, towards the sigmoid colon, and it's going to be stored there until we are in a position to defecate. So what makes this retrograde movement possible from the rectum to the sigmoid colon backwards? How is it possible? And this is because the BER, or the basal electrical rhythm, in the rectum is higher than the sigmoid colon. So the slow waves of contractile activity that we have is higher in the rectum than the sigmoid colon. So this causes the pressure in the rectum to be higher than the sigmoid colon, and obviously this pushes it back into the sigmoid colon if needed. I don't know if you guys remember that one diagram, so I will go back till I find it. Okay, here we go. In the rectum, it's 17, and in the sigmoid colon, it's less than 7. So you can see there's a difference, and that's why it can move backwards this way. Okay, so this is a quick review. So in the ascending colon and the proximal transverse colon, we have segmentation, non-propagating, haustral, shuttling. All of these are the same, they're just different names. So we have this type of contraction, okay? But in the distal transverse and sigmoid colon, we have peristalsis and propagating contraction. This type of contraction is good for mixing and for absorption. This type of contraction is good for moving it towards the rectum. Okay. This causes actual movement, this one. This doesn't cause any movement, it just causes mixing. So why is that useful? So segmentation or non-propagating contractions are useful because it allows for efficient absorption of electrolytes in water. Since it's slow, it gives us time for the mixing to happen and also gives time for the absorption to happen. And that's why it being slow is an advantage, not a disadvantage. And obviously it's slow due to low BER and no BER gradient. Okay, no BER gradient. Both of these factors play into it being slow, and we discussed this in one of the slides earlier. Okay, so we're done with 10 slides now, or this is the 10th slide. So does anyone have any doubts? You can ask me. Just unmute your mic and ask me. If there are any doubts in the chat, you can read it out to me because I can't see the chat right now. Before I move on to the next part. Okay, so this is a really easy slide, and it's just about the colon transit time, which is the amount of time the feces spent in the colon. And obviously this depends on your diet. If you have a diet that's good, an ideal diet that's rich in fiber, then it will be lower. And if you have a diet that mostly consists of like non-ideal food, including a lot of processed food, then it would be higher. So for average Western diet, which is what we consume, it's 1.5 days on average, and the maximum is 4 days. Longer than this would cause constipation, obviously. The colon transit time is faster in men, and men also have increased fecal volume. So this is obviously due to the fact that usually men eat more than women to consume more calories per day, and this causes an increased fecal volume. So adding fiber, such as bran fiber, reduces the colon transit time because it makes it easier for, it basically provides lubrication for the feces and allows it to pass through the colon easier. And we have this graph over here. Non-bran is black and the bran one is white. So you can see the transit time is over here on the y-axis, and it's in hours. So you can see the higher hour ones are all non-bran, and then when you get to the lower hour ones, such as this, they're all bran. So basically, it's telling us that bran reduces the colon transit time, which is good, and you'll see why it's good later, because the colon transit time tells us which kind of nutrient is going to be fermented. And we'll learn more about this in one of the next slides. But just remember that fiber is good and reduces colon transit time. I can't move to the next slide. Just give me a second. Okay, so now we have colonic fermentation, which I was talking about earlier. And we have bacteria in the colon. We have bacteria all throughout the GI, and similarly, we have them in the colon. And they prefer fermenting carbohydrates. So for them, it's ideal if we have carbohydrates, right? However, if carbohydrates are depleted, they can also ferment proteins. So they can ferment carbohydrate, which is their first choice, okay? But they can also ferment proteins, which is their second choice. However, they will not ferment lipids in physiological conditions. And the reason for this is lipids are absorbed in the small intestine via micelles, right? So technically, there shouldn't be any lipids in the colon in the first place, and that's why they will not ferment lipids. But in pathological states, such as in pancreatic enzyme deficiency, when you have deficiency of lipase, or decreased bile pool salts, which help in the formation of micelles, in these cases, lipid absorption is going to be affected, and this is a pathological state. And in these states, lipids will be present in the colon, and then the bacteria can ferment lipids. But usually this does not happen. Carbohydrates are their first choice, and proteins are their second choice. Usually lipids are not a choice at all. So now, if we have a short transit time throughout the GI, okay, and obviously this can be achieved by eating fiber and having a well-balanced diet, this favors carbohydrate fermentation. And if we have a prolonged transit time or a longer transit time, obviously this can happen if you have an unideal diet, if you do not have fiber, if you drink less water. This all increases the transit time because it decreases lubrication in the colon, which makes it harder for the feces to pass through. This favors protein fermentation. And the reason for this is, if the transit time is prolonged, the carbohydrates will be absorbed by the small intestine, okay, leaving nothing in the lumen. So all the carbohydrates are going to be absorbed in the small intestine, and whatever feces goes to the colon, there's going to be nothing in it except some proteins. And then the bacteria are left with no choice but to ferment proteins, right? Now, you may be asking why this is important, and the reason for this is carbohydrate fermentation is considered beneficial to health, but protein fermentation could lead to toxic metabolic byproducts, okay, such as sulfides, okay, such as amines, and other potential carcinogens. So ideally, we do not want any protein fermentation, and obviously we do not want a majority protein fermentation, which is what would happen if we have a prolonged transit time. This is why people recommend eating fiber, because that decreases the transit time and favors carbohydrate fermentation. So protein fermentation is bad because it causes toxic products and potential carcinogens to be formed. We have something called prebiotics, which includes like yogurt, and there are also supplements that are prebiotics. These are fiber rich. Obviously, this is a good thing, because this reduces colon transit time, which means we'll favor carbohydrate fermentation, and it maintains healthy microbiota. So it doesn't allow the microbiota balance to be harmed, because if we have the wrong microbiota in the colon, that also affects us just as much as if we have a fiber-rich diet or not. So it does both things, prebiotics, and that's why they recommend it. Okay, so now we move to this thing. It's really simple. There's two that you need to focus on. This is the first one. There's two transporters. This is the first one, and it's called ENAC. Okay, the full form of this being epithelial sodium channel. Okay, and then there's this one that you need to focus on. Oops, sorry, this one, not this one. So this one is a normal potassium transporter, ATPase, that you guys know of. It's present everywhere in the body. So this is the lumen, this one, and here's the interstitium. This is the cell of the colon. So in the lumen, we have the feces, right? And this feces contains water and some electrolytes, and one of these is sodium. So obviously, you want sodium to be reabsorbed. You don't want it to be excreted in a large amount. So we have this thing called an epithelial sodium channel, which is this thing. And this basically is present in the distal colon, and this absorbs sodium into the cell. Now, we don't want this sodium to remain in the cell, right? We want it to return to circulation in the body and go wherever is necessary. So to remove it from the cell and put it into the interstitium towards the blood vessels, we have this transporter on the basal membrane of the cell, and this is a sodium potassium ATPase, which exchanges sodium for potassium. This is a very common one. And along with sodium, you obviously have water coming through, because water is going to follow the osmotic gradient that is created by the sodium. So water is also going to move into the cell. And both of these channels, the ENAC and the sodium potassium ATPase, are regulated by a hormone, called aldosterone. Wait, actually, I need to search up if it's a hormone or not. I'm pretty sure it is. If anyone knows, if anyone remembers, actually, you can tell me. It is a hormone, though, I think. Yeah, it is a hormone. Okay. Phew. Aldosterone, you'll study more about it in the endorepro block. So aldosterone stimulates both of these, to absorb sodium and exchange sodium, respectively, so that sodium can return to the bloodstream. And aldosterone, as you will learn later in the endoreper block at the end of this year, basically wants there to be an increase in sodium absorption. So just remember it for now. Aldosterone stimulates both of these ion channels and transporters to increase sodium reabsorption. And obviously water follows because of the osmotic gradient. I hope that's clear. Now, you remember that one slide where we talked about mass movement, where I said usually the movement of the feces towards the rectum is slow, except if we have mass movements. And I told you to keep it in your mind because we'll study about it in a bit. So here we have the slide about mass movements. And these are not controlled by the intrinsic slow waves or the BER, such as, which was the case for the colonic segmental slash haustral slash non-propagating contractions. Those were controlled by the BER, or the basic electrical rhythm. These, however, are regulated by the autonomic nervous system, more specifically by the enteric nervous system, which is a subdivision of the autonomic nervous system. So these are regulated by the autonomic nervous system, not the basic electrical rhythm, which is through a pacemaker present in the GIT. So its peristalsis is similar to the small intestine. I'm sure you guys already done that. But there's a difference because the mass movements in the colon can move contents or feces up to 20 centimeters along the colon, which is 10 times more than the small intestine, where in the small intestine, peristalsis can move at 2 to 3 centimeters, but in the large intestine, it's up to 20 centimeters in the colon, right? This mass movements occur 1 to 3 times a day in healthy humans, and they usually begin in transverse colons, and they move the chyme to the rectum in a fast manner. So I told you guys earlier, it's fast, but it's usually slow, sorry. Most cases it's slow, but if you have the mass movements, then there's a fast movement from the transverse colon to the rectum. And it's usually absent in fasting individuals. So this only happens if you've eaten. So as it says over here, but it occurs after ingestion of meals and precedes and accompanies defecation. So it happens, number one, when you ingest, and number two, while you're defecating. And the reasons for this obviously is during the ingestion of the meal, so that it can move the previous meal, the one before this one. It can move it away from the colon to accommodate the new meal, and during defecation, it's so that all of the feces that are present in the colon are emptied out, right? But while you're fasting, it doesn't happen. Okay, so we're done now for this part of the lecture. So if anyone has any doubts, you can ask me now. I think we should take like a 5-minute break to go pray and then come back. Is that okay with you guys? Is that okay with you guys? We can pray and then come back in 5 minutes. Okay. Okay. So we're gonna continue where we left off. So we're done with the colonic peristalsis. Oops. We're done with the colonic peristalsis, so we're gonna move to the next part, which is the rectal motility. So now we've done everything. We've passed the food through every part of the large intestine and it's reached the rectum. And in the rectum, we have something called goblet cells, which are also present in other parts of the body, but they're present here. So these goblet cells produce mucus, and this mucus lubricates the surface of the rectum and eases the passage of semi-solid stools. So it makes it easy for the feces to be moved into the anus. It lubricates the walls of the rectum and the feces. So the exit of fecal material is controlled via smooth and skeletal muscles. So as you know, smooth muscles are involuntary and skeletal muscles are voluntary. So we have both of these muscles contributing towards the movement of fecus into outside our body, right? So the smooth muscle is called the internal anal sphincter, which is this one over here, internal anal sphincter. And then the skeletal muscles, there's two of them. The first one is the external anal sphincter, which is this. And we also have the pure rectalis muscle. So I'm going to underline them over here. So we have both smooth and skeletal muscles controlling the exit of the fecal material. The pure rectalis creates a sharp angle between the levator ani muscle, which is another muscle, which you'll also study later in the year in endorepro. But just for now, remember, just it creates a sharp angle between the levator ani and the external anal sphincter, which helps to maintain the fecal continence, and this is known as the fecal continence is basically the fact that when we voluntarily stop the excretion of feces, because we're not in an ideal situation for excretion, that means the feces is stopped, and it's either stored in the rectum or it moves through retroperistalsis into the sigmoid colon, right? So this is called continence, and there is a definition of it in the next slide, I think. But yeah, pure rectalis creates the sharp angle between the levator ani muscle and the external anal sphincter, which maintains fecal continence, right? Both the sphincters are tonically constricted, and this is an important thing. So both of them, that is the internal sphincter and the external sphincter, are tonically constricted, which means they have a tone to them. In the resting state, they have a tone to them, which means they're always contracted until and unless a specific signal is given to them, which causes them to relax. Okay? And this specific signal is the defecation reflex, which we'll study about after this. Like, there's a whole slide for the defecation reflex. And also, we have, we studied about accommodation and retroperistalsis earlier. Accommodation is when the feces is stored in the rectum, and retroperistalsis is when it's stored in the sigmoid colon. And this is obviously when we voluntarily stop the excretion of the feces. Suppositories are medications that are administered via the rectum, right? And this is very common in babies who cannot swallow pills. So you give the medication to the rectum, to the anal canal, and then it's absorbed because this area is very well perfused with blood vessels. There's a lot of blood vessels here. So this medication is absorbed into the blood vessels and then taken into systematic circulation. So yeah, tonically constricted is a very important point. They're always constricted until we have a specific signal that tells them to relax. Okay, and this obviously helps in continence. We'll say more about the signal later. And here's the signal. We have an intrinsic reflex and an extrinsic reflex. So first of all, we'll study about the intrinsic reflex. The external anal sphincter is a skeletal muscle and it's voluntary, right? So if it's inconvenient, that means we're not in a condition to defecate, there will be voluntary contraction of the external sphincter via the pudendal nerve. So we have a nerve called the pudendal nerve that supplies the external anal sphincter. And if we're in like an inconvenient condition where we do not want to defecate, then the voluntary contraction of the external sphincter will be via the pudendal nerve. Okay, and this blocks excretion because it closes the sphincter, right? If it's convenient, there will be voluntary relaxation of the external sphincter, causing defecation. Okay, so if it's convenient, there will be relaxation. If it's inconvenient, there will be contraction via the pudendal nerve. So so this is the case for the external sphincter, but what about the internal? Because that's involuntary. It's a smooth muscle, right? The internal anal sphincter. So if we have mild distension of the rectum, it results in a passive increase in rectal pressure because we have something pushing against the rectum. If the distension is enough, the passive increase in pressure is further increased by active contraction of rectal muscles, okay? And this is when we feel the urge to defecate, right? And the urge increases as the rectum continues to fill. So as you keep filling the rectum with more feces, the urge increases, okay? And this will lead to relaxation of the internal sphincter. So when you have this rectal pressure, which is further increased by the contraction of rectal muscles, this leads to relaxation of the internal sphincter via the parasympathetic reflex interacting with the enteric nervous system that is mediated via NO and VIP. So that is a lot to take in at once. So let's break it down a little. So we have a parasympathetic reflex system. So parasympathetic is a part of the autonomic nervous system. And obviously, I don't know if anyone's ever mentioned this to you before, but excretion is parasympathetic. In fact, all of the activities in the GIT are parasympathetic, because parasympathetic is when your body is relaxed, right? And obviously, all of this stuff only happens when you're relaxed. If you're stressed, such as in the sympathetic system, this is not going to be happening. So when the internal sphincter is opening, it's via the parasympathetic system interacting with the enteric nervous system that has control over the GIT, which is also part of the autonomic nervous system. It is mediated via NO and VIP. NO is nitric oxide and VIP is vasointestinal peptide. Both of these are very strong relaxants, okay? You've probably studied of them in foundation, and basically they lead to relaxation of smooth muscle. And as we said earlier, the internal sphincter is a smooth muscle. So the NO and VIP are released via the ENS because of the parasympathetic system, and it relaxes the relaxes the smooth muscle, causing the internal sphincter to relax, okay? So this is how the internal sphincter is regulated and how the external sphincter is regulated. So we have a diagram here that's really helpful. So this one is a rectum. This is the pressure in the rectum. This is the internal anal sphincter and the external anal sphincter. So in the rectum, we have a passive increase in pressure over here because it's filling with feces. And then here we have an active increase, which means the rectal muscles are contracting. Obviously, when there's active increase, there should be a decrease in internal anal sphincter pressure. Why? Because the internal anal sphincter is relaxing, which means the pressure is going to decrease. The pressure was increased when it was tonically constricted, but now that there's active contraction of the rectal muscles and passive pressure because of the feces, it's going to relax, and this is going to lead to this decrease in pressure shown here. And obviously, you see as this increases, as the pressure increases because of more feces and more contraction, the internal anal sphincter pressure will decrease. However, you see the external anal sphincter is not; it's still it's still contracted. There is no decrease in pressure, and this is due to the fact that obviously it does not care if there is pressure in the rectum because of the feces. It's only controlled by the pudendal nerve, and the pudendal nerve it's obviously a skeletal muscle, so it only cares about voluntary control. So unless your brain tells it to relax, it's not going to relax, and that's why you see that there is no decrease in pressure unless the brain wants there to be, as you see in this case over here. But this is controlled by you because it's voluntary. So the external sphincter contracts in response to The external sphincter contracts in response to rectal distension. So as I said earlier, the internal sphincter is going to relax when you have distension of the rectum, right? Because of the VIP and the NO and the PNS and the ENS, right? Like all of those together. But the external sphincter is going to do the opposite. It's going to contract. And it will contract via extrinsic somatic reflex, which is activated by stretch receptors in the rectum through the pudendal nerve. So let's break that down because that's a lot of words. So the extrinsic somatic reflex, somatic means something that's controlled by you, right? Activated by stretch receptors in the rectum through the pudendal nerve. So basically, this is the rectum, okay, and you have feces in it. It leads to pressure on the walls of the rectum, right? We have stretch receptors that activate the pudendal nerve to contract the external anal sphincter, okay? And that's basically what it's saying. It's saying that if there's feces in the rectum, it causes contraction of the external sphincter. However, it causes relaxation of the internal sphincter. And the external sphincter only relaxes when you want it to, because it's voluntary. So when your brain gives it a command to relax, then it relaxes. Okay, just like any other muscle. Like if you flex your bicep, and then you relax it, When you give your brain the order to relax it, that's when it's relaxing. It's the same thing for this external sphincter. And if the urge to defecate is opposed by the voluntary contraction of the external sphincter, then the rectal receptors eventually adapt and the internal sphincter regains tone. So what does that mean? So that means if we're not in an ideal condition to defecate, then obviously the voluntary contraction will cause closure of the external sphincter, right? The external sphincter is going to be closed. And eventually, if it's closed for long enough and the feces cannot pass out, then the rectal receptors, which are within the rectum, are going to adapt. They're going to understand that, okay, we cannot currently defecate. So what it does is it tells the internal sphincter to regain its tone. So remember how both the sphincters have tonic contraction, right? So it's going to tell the internal sphincter to regain its tone because they cannot defecate right now. Like, it has understood that you can't defecate right now. Subhanallah. So basically, let me repeat that again so that you guys understand. The internal sphincter will regain its tone. That means it will contract again once it understands that you cannot excrete. And how does it know that you cannot excrete the feces? It's because there's voluntary contraction of the external sphincter, which is controlled directly by your brain because it's a skeletal muscle, right? And we've mentioned the word continence a lot. So here's a definition. I already mentioned it earlier, but I'm mentioning it once again. It's the ability of the individual to override the urge to defecate by voluntary contraction of the external sphincter. So even if you want to defecate, if you have feces in your rectum, but you're not in an ideal condition, then the external sphincter is going to contract and not allow the feces to be excreted. And that is what continence is. Okay. So this is a quick review. I want you to focus on this part for now, although we will talk about this as well, but focus mostly on this. So we have distension of the rectum due to the feces being accumulated here. Okay. And this causes relaxation of the internal anal sphincter. It leads to a loss of tone. And it leads to a contraction of the external anal sphincter, okay, via the pudendal nerve because of the stretch receptors present in the rectum. All right. Now we have this part. So this is one part. This is the short pathway, okay, also known as the intrinsic pathway. And then this pathway over here is the extrinsic or long pathway. So as we said, if it's convenient for you, then the voluntary external anal sphincter is going to relax and is going to lead to defecation. And this is obviously under voluntary control, and it's going to lead to the relaxation of the external anal sphincter. If it's inconvenient to you, then the external anal sphincter is going to constrict. It's already constricted over here, as you can tell, but it's going to constrict further. And the reason for this is because in the short or the intrinsic pathway, when the external anal sphincter constricts because of the stretch receptors, it is just transient. Okay? So while it does constrict, yes, it's transient, but if your brain tells the external anal sphincter that you cannot defecate right now, then it's going to constrict permanently, and this is what we call continence because it's under our control, and this leads to further constriction of the external anal sphincter. I know this is a bit confusing, so do you want me to repeat this, or did you guys get it? Because I know it can be confusing, but it's not that hard at all. Like, it's just the first time might be a little difficult, but other than that, it's not really that difficult. So does anyone want me to repeat it? If you want me to repeat it, you can unmute your mic and say yes. Otherwise, I'm going to continue. Okay, I guess no one can repeat it. Okay. So now we talked about the intrinsic reflex. This is the extrinsic reflex. It's also related to the intrinsic reflex, so it's all very well regulated. So the defecation reflex is reinforced. The defecation reflex being the intrinsic one is reinforced by the extrinsic reflex, which is mediated by pelvic nerves. So the extrinsic reflex is via pelvic nerves. Okay. The afferent signal is from the rectum. So once the rectum is filled up with feces, obviously there are stretch receptors, and it's going to send an efferent signal with an A. Okay, this efferent via the efferent nerve fibers is going to send a signal here to the spinal cord. It's a sensory signal telling the spinal cord that the rectum is full. Okay, then we're going to have an efferent signal with an E, okay, via the pelvic nerves to the descending and sigmoid colon rectum. Okay, so the blue one over here, this one going here, 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 and here. So to the descending sigmoid, the descending colon, the sigmoid colon, and the rectum, and it basically causes contraction and relaxes the internal anal sphincter. Because if the rectum is full, obviously that means we have enough feces and we want to defecate now, right? So it causes further contraction of the descending sigmoid and the rectum. Further, we have an efferent signal from the pudendal nerve, which causes relaxation of the external sphincter, which is this one, the red one. Okay, the pudendal nerves coming from the sacral segment. You don't need to remember that, just remember pudendal nerve for now. Pudendal nerve also has an efferent signal, efferent with an E, okay, this one, which causes voluntary removal of tone. Okay, so basically, I hope you guys have understood by now that this slide is basically showing us if we do want to excrete, if we do want to defecate, then how does it happen? So the first thing is we have the afferent signal from the rectum, then the efferent signal from the pelvic nerves causing contraction of the descending colon, sigmoid colon, and the rectum. Then we have an efferent signal from the pudendal nerves causing relaxation of the external anal sphincter. And then when all of this has happened, then we can excrete. We also have an efferent signal via the sacral nerves to cause relaxation of the puborectalis, which is the other muscle that's a skeletal muscle and also helps in continence, right? So once all of these have relaxed and we have contraction, this is when we can have defecation. Obviously, the external one is under voluntary control, which is the one controlled by the pudendal nerve and the ones controlled by the sacral nerves, that the puborectalis, they control the puborectalis. And the pudendal nerve controls the external anal sphincter. So now I'm going to ask you a question. You already have the answer on the screen, but what prevents involuntary defecation following sudden increases in intra-abdominal pressure, such as what's caused by sneezing, coughing, lifting heavy objects? So I know the answer is already on the screen, so I'll just answer it myself. It's due to the tonic contraction of the external anal sphincter and the puborectalis. Remember, these can only be relaxed, okay, it's under your control. And they are relaxed via the efferent signals through the pudendal nerve and the sacral nerve, right? As is mentioned in this slide, okay, these two. And this is when it relaxes. And when it relaxes, obviously, you can defecate. This only happens when you're in an ideal situation to defecate. Okay. Now we have something called Valsalva maneuver, and it was mentioned in the notes, and that's why I mentioned it over here. It's basically forced expiration against the closed glottis. I mean expiration, but without expirating. Like you're doing the motion, but you can't actually expire the air. And you're choosing to do so because it increases intra-abdominal pressure and lowers the pelvic floor. Okay. We also have something called the anorectal angle. Okay. And this is basically an angle that's between the anus and the rectum. It's present anatomically, so it's present there, and it prevents incontinence because of the angle, it prevents incontinence. And when we sit or squat, it decreases the anorectal angle, which makes it easier to defecate. And this is why you if you have the urge to defecate and you sit down, it furthers the urge because the anorectal angle is decreasing, which is the anatomical thing that was stopping incontinence is being removed because the angle is decreasing, and this causes the urge. It causes an increase in the urge. So also squatting further decreases the angle. So sitting and squatting decrease the anorectal angle, which makes it easier for us to defecate. So this diagram over here is very simple. It's just saying that if we have this anorectal angle and the puborectalis and the sphincters, if they're all constricted, and you're standing, then basically the feces, even if it is in the rectum, it has no chance of being defecated because of all of these preventative measures. If we sit or squat, then the inner rectal angle is going to be decreased. And then if we give an order via our brain to the external anal sphincter and the puborectalis, because both of those are skeletal muscles, to relax. Now, once those are relaxed and we have feces in the rectum, and obviously if you have feces in the rectum, that means the internal anal sphincter is going to be relaxed, right? Because of VIP and NO and ENS and PNS, right? So this is going to cause the feces to be defecated, and then eventually defecation is completed. I hope you guys remember what I'm talking about. I'm talking about this right here. So here, internal sphincter is, first of all, if you have rectal pressure, passive due to the feces. Second, active pressure due to rectal contraction. Both of these lead to relaxation of the internal sphincter. That's what I was talking about over there. That's already relaxed internal sphincter via the ENS and the nitric oxide and vasointestinal peptide. So we're almost done. There's like 2 slides left. So this quick summary. We have a mass movement of the feces, and it reaches the rectum, and this causes a defecation reflex. The stimulus is the distension of the rectum by the feces. The response is the contraction of the rectum and the sigmoid colon, and this contraction obviously is due to the afferent signal from the rectum and the efferent signal from the efferent signal, the pelvic nerves, right? So this whole pathway, the afferent and the efferent, like this. That causes contraction of the rectum and the sigmoid colon. We have relaxation of the internal anal sphincter due to the pressure on the rectum, which causes the release of VIP and NO. We have contraction of the external anal sphincter, right? Because when we have a defecation reflex, the external anal sphincter contracts transiently, okay? By the pudendal nerve, because we also have stretch receptors in the rectum that are connected to the pudendal nerve, and once those are activated, it causes the pudendal nerve to cause the external anal sphincter to contract. So obviously, all this increases the pressure in the rectum, and if we delay the defecation, that means if we're not in an ideal condition, then we have contraction of the external anal sphincter via the pudendal nerve, which causes also contraction of the puborectalis via the sacral nerves, and this causes retroperistalsis in the rectum. It pushes the feces back to the sigmoid colon for storage until the opportunity presents itself to defecate. But if we're in an ideal situation to defecate, it causes relaxation of the external anal sphincter, which is completely under your control. You send an efferent signal, E with an EFF, ERNT, efferent signal to the external anal sphincter and the puborectalis muscle through the pudendal nerve and the sacral nerves, respectively, which causes relaxation. We also have forward peristalsis in the rectum and the sigmoid colon because of the afferent and efferent pathway, and you have the Valsalva maneuver, which I told you earlier, that increases abdominal pressure, where you breathe, you expire against a closed glottis that increases intra-abdominal pressure, which pushes the pelvic floor down. Actually, you can notice yourself doing this if you have, if you've ever noticed yourself having constipation or something like that, you will unconsciously do the Valsalva maneuver to make it easier for you to excrete your feces, because it causes the pelvic floor to lower, it reduces the anorectal angle, it increases abdominal pressure. All of this makes it easier for you to excrete your feces. Okay, so constipation is obviously when you have hardened feces, and this is due to poor motility in the gastrointestinal system. If you have poor motility, it's going to increase the transit time. If you have an increase in transit time, you're going to have greater absorption of water and solutes, leaving us with hard feces in the transverse colon, which causes constipation, right? Pretty simple. Now, these 2 diseases, they're the last 2 slides mentioned in the lecture. I don't know if you're going to be tested on them, because usually you're not tested on diseases in first year, but I still decided to put them here. So achalasia is also called It's called mega esophagus. So you have to go all the way back to the start of GIT to know this. You have something called the lower esophageal sphincter, right? And it is also tonically constricted. So it's usually constricted, right? Because we don't want random things to enter our stomach via the esophagus. So now, either due to an autoimmune disease or due to some viral infections, you're gonna have the inhibitory neurons are gonna be destroyed. So these inhibitory neurons usually release VIP and NO to relax the lower esophageal sphincter so that food can enter. But if these inhibitory neurons, let me write it down here, inhibitory neurons are destroyed, then obviously this will not relax. It'll stay constricted. And this makes it harder for us to swallow food. It leads collect over here. So I'm going to go into the stomach. You're going to see a narrowing of the lumen, as you can see over here. Usually the lumen is larger. And the most diagnostic feature is this bird's peak on an X-ray. So basically, we give the patient a barium swallow, and then we do an X-ray. And then you see this characteristic bird's peak, right? Which looks like the peak of a bird. And this is because all of the barium is like collected over there because the lower esophageal sphincter is constricted and it's not going to allow it to enter the stomach. And this is called achalasia or megaesophagus, right? So yeah, that's it for this disease. And then we have something called Hirschsprung's disease, also known as congenital megacolon. So if you guys remember something called neural crest cells, which are present, which like disperse to different parts of the body during during development. I think you guys might have done this in the foundation block. The neural crest cells are also present in the walls of the large intestine. So in some cases, we have these neural crest cells, they do not migrate correctly into the walls of the colon. And basically, we lose the parasympathetic activity for peristalsis and all of this stuff. So this causes constriction of the colon. As you can see in this case, this constriction is due to the loss of parasympathetic, the parasympathetic system and the enteric nervous system because the neural crest cells don't migrate. This causes constriction here, and all of the food is collected, and this causes dilation backwards, like distally, and this is basically called congenital megacolon. Megacolon because you have dilation, right? And the affected part is actually the constricted part because this is the part where we have loss of parasympathetic innervation. And yeah, that's Hirschsprung's disease. You might have also done this at foundation. I think we did it in foundation too. Yeah, that's it. We're done with the lecture. You can scan this code for the feedback. It's literally just 1 or 2 questions, so it'll be really helpful if you could answer. And inshallah, if you answer, I guarantee you an A in GIT. So if you have any questions, you can ask me now. We're done. I'm here to explain anything that you want. And yeah, thank you for attending. If you have any questions, you can ask me now or you can ask me later, whatever. I'm here. But yeah, we're done. Thank you so much.